Welcome to this MI4 celebration. Thank you very, very much for being here. I think that many of you in the room were here in 2018. Some of you have joined us along the way since then, and what I'd like to think is most of you are here to stay and help us celebrate this event, and hopefully we'll be here to stay to celebrate many more events to come. I'd like to welcome three very special people in the MI4 community. Two of our staunchest supporters, the Vice Principal and Dean of the Faculty of Medicine, Dr. Eidelman and Heather Monroe-Bloom. You have supported us, you have advised us, you have guided us, you have been tremendously important in shaping what MI4 has come to be. Thank you very much for being here. I'd like also to point out the presence of Monsieur Paul Marchand, who is with us. Monsieur Marchand was really responsible for the foundational gift that really launched MI4 in 2018. Without that gift, we would not be here. And again, Monsieur Marchand, I don't know how else to thank you, but to thank you very much sincerely. J'aimerais présenter une reconnaissance de terre, land acknowledgement. Je pense qu'avant de faire cette reconnaissance-là, je pense que c'est important de mentionner que beaucoup des maladies qui nous intéressent à MI4 sont représentées de façon disproportionnée chez les peuples autochtones. Alors je pense que c'est land acknowledgement, cette reconnaissance de terre est particulièrement pertinente pour cette réunion aujourd'hui. Alors l'Université McGill est sur un emplacement qui a longtemps servi de lieu de rencontre et d'échange avec les peuples autochtones, y compris les nations Haudenosaunee, Anishinaabe, Huron-Wendat et Abenaki. McGill honore, reconnaît et respecte ces nations dont la présence marque le territoire sur lequel nous nous réunissons aujourd'hui. Et finalement, I'd like to make this a little bit personal. So I was not here in 2018. I was actually, Marcel and Don started MI4. They approached me in 2019 to come along. I like to say that they thought that I could represent a little bit of diversity in the leadership. And I remember that initial conversation with Marcel and Don when they said, you know, would you like to join us? And I said, no way, this is not going to happen. I can think of 1,000 reasons why I'm not the right person that you're looking for. And Marcel at that point said to me, well, Marie, you can say what you want, the dean is going to call you. And so the dean called me, and the rest is history. And so I'd like to take this opportunity to thank the dean for having nudged me in the right direction. This has been really a great opportunity for me to serve my university, and I'm really grateful for that. Thank you very much. So on that, I'd like to introduce our dean to address you for a bit.
Well, thank you very much, Marie. I have to say, I remember that conversation very, very distinctly. It was on the telephone. I was in my office holding the phone and pacing back and forth and trying to think of cool ideas and reasons why you should do this. So I'm glad I succeeded. So as many of you know, I am in the twilight of my deanship, and Leslie Fellows will be succeeding me on August 1st. And I'm in that role, in that period of time, I'm thinking a lot of what happened while I was dean. A lot of things happened. Things are still happening. But one of the best things that happened was MI4. Because it's really an exceptional example of what can be accomplished when people have good ideas and then they put them together with collaboration and with a sense of community that goes far beyond the usual. I'm used to, I see several of my fellow Meekins Christi people here. We're active in MI4, and we have a great familial feeling in the Meekins Christi lives, but it's not the same thing as something that encompasses the entire university community on all our campuses. This is really unique. And it also, of course, the timing could not have been better, so that made us look really smart. But I'm most proud of it because of what it did to build and reinforce a collaboration and cooperation across the university, allowing McGill to take its rightful place as a world leader in these areas. I also think it's allowed us to see amongst the people in this room, people who've risen to leadership in different projects in different areas that maybe most of us wouldn't have heard of unless we happen to be in those lab groups. And I think this is also a great thing. Also, as was mentioned, it allowed us to reinforce our collaboration with between the faculty and the university and our teaching hospitals, especially the MUHC. And it was, as was mentioned, Mr. Marchand and the Doggone Foundation and their fabulous support were instrumental in moving this forward, but also, again, reinforcing the sense of community. So MI4 is a real success. It's wonderful to see it make it to its 5th anniversary. I can tell you that after MI4 started, I had a parade of people in my office who wanted to start MI4-like entities in every topic you can imagine. Cancer, stem cells, you name it, there was somebody who wanted, and they would come in and they would say, you know what we need to do in my area is we need an MI4 for this. Well, this is the MI4 for MI4. It's by far the most successful. It's by far the most dynamic and most exciting. And I look forward to seeing what happens. I've been very impressed with what happened in the last 5 years, but I'm very excited about what's going to happen in the next 5, 10, and 15 years. So congratulations on making it to your 5th anniversary, and thank you for allowing me to be a part of it. Thank you.
Good afternoon, everybody. I can't believe that I couldn't manage to make my schedule work to be there to celebrate with you all in person. I'm very disappointed. But let me start with the most important thing, and that is congratulations to all of you. 5 years out, MI4 is alive, well, and thriving. I can't believe that we got here. I have vivid memories of multiple milestones along the way. Conversations with Marcel in our offices about crazy dreams of what we could do to kickstart infection and immunity. That first meeting we had at the RI where somebody suggested the multiple I's that became MI4. That first town hall symposium we had trying to figure out what kind of vision we could put into place, what we could accomplish, dreaming big, blue sky, putting people together, and look where we've landed. The last 5 years have been an incredible ride for me, and I know for Marcel and Marie, and I'm sure for everybody else in the group. We're on a great path. We have the resources. We have an incredible community that is invested, interested, and oh so creative. And I can't wait to see what the next 5 years is going to bring. Happy anniversary.
All right, let's see if I can get this to my height. Can everybody hear me in the back? Okay, great. All right, so I've been asked to give a little perspective, a look back and a look forward at 5, and I guess the idea is to try to project where we're going to be in 2028. So first of all, thank you all for being here. Thank you, Heather, David, Paul, and all the scientists who were at all the meetings, the discussions, the breakout sessions who've created the vision and the action that became MI4. So I'm going to do a look back. This is what you see off the back of my sailboat when you anchor it just off here in Lac Saint Louis and you're watching the sunset. You see a few other boats that are floating around. And then the look ahead. Where's Kendra? This is when we went out on an MI4 sailing evening and she's looking into the future and seeing where the boat is going. And in between, I'll briefly take stock of where we are right now. So here's the timeline. The meetings that Dawn refers to, the concept really came around 2016, 2017. And a lot of this came as the result of the merger of the MUHC hospital where the Meekins and the Duff and the Montreal General and the Children's and all these groups were coming together and there were new collisions. And we said, well, if we saw what happens with new neighbors and collisions and new collaborators, what if we just grow that, take the same concept and make it bigger? The launch gift from the Doggone Foundation 5 years ago this week We started to have programs in 2019. These evolved very quickly into COVID programs in 2020. I was asked to be the new director in 2021, and then in 2022, we had a whole series of discussions with members of external advisory board like Galit and stakeholders who were in the room here, members of the centers, about making sure that we were going to be pertinent for the next 5 years and what would be our focus and how could we keep MI4 going from excellence to excellence. And here we are turning 5. I look back in the past few years and I can find slides from our slide deck. They're all on the Google Drive that David has told me us academics are not supposed to use because we're supposed to use SharePoint, so hopefully this wasn't recorded. But at least I can find things on the Google Drive on most days of the week. Here's a slide that we put out around 2018, 2019 saying Zika bites. And you could see how a virus could spread around the world. And we were saying, look, something is in Brazil in 2014, and it's in Florida in 2016. And we had all these flight maps, and now we know, of course, that other viruses can fly around the world pretty quickly. We'd identified a new pandemic and antimicrobial resistance as the first major threats. And two of the priorities we identified then were containment labs and clinical research platforms. Here's a slide that Erwin Schur, where's Erwin? He shared with me from a meeting that he attended with Marie, and he told people that these containment level 3 labs, depicted here, offer a safe and secure environment to keep the harmful pathogens on the inside so that you can share your discoveries with the outside. That idea that a platform like that could be enabling for all the scientists in the room was sort of an idea. In 2019, we got somehow something else seemed to have jumped on that. I don't know what happened there. Anyway, we got 400,000 from the MUHC Foundation, which became 7.2 million from the Canadian Foundation for Innovation, which is an 18-to-1 multiplier. And then these labs then were the place where papers from Maz's group or Nahum Sonnenberg's group with Sylvia and others then were able to work with SARS-CoV-2 and publish their findings about certain aspects of the biology of infection. And they're continuing to be used now for tuberculosis and SARS-CoV-2, and they should be ready for the next pandemic. So what was another slide that we were showing? This came from one of the Dragon's Den events. So in 2018 and 2019, we'd get people together in a restaurant and ask them to give us their craziest ideas, and we'd have 3 people sit there pretending they were on a TV show, and one would be a lawyer, one would be a businessman, and one would be from maybe the Concordia Innovation Program, and they would say, tell us what's your big idea and what makes you so unique. And Marina Klein put up this idea. She said, we should have a clinical trials platform. Because the idea is that to go from having an idea in the top left and saying, this is what I want to do, and going through the getting money and writing the protocol and finding a data secure system and taking the samples and then analyzing the data and making sure the data is secure and regulatory compliant and getting to New England Journal article can be an impediment. But if we provide this pipeline already, then when somebody has the idea, a lot of the other steps have been smoothed, if you like. And we saw that this clinical trials platform became enabled during SARS-CoV-2, of course. We saw that some of the samples from this clinical trial platform have already led to a patent which brings royalties back to McGill University for a cell-free DNA technology led by Matt Cheng, and I think Don Vin was involved in that work as well. But then that platform became a legacy because even when SARS-CoV-2 was spreading but COVID was abating, this platform was the place that was able to recognize monkeypox, now called Mpox. So here's Safa Barkati. She identified the first cases in Montreal with Marina Klein. She collaborated with other scientists here and around Montreal and around the world. And there's their New England Journal article, which was as Marina had predicted in 2019. So those are several of the success stories, and if I take stock in April of 2023, we can say that when we look at the funding brought in by McGill University Advancement and the MUHC Foundation, so far the direct funding has reached around 40 million, and then of course there's the leveraged funding, things like the CFI, and then you can even try to count all the CIHR grants and all the other grants that came off of these capacities. Have we had successes? Yes, I think we've had successes. Have we had failures? Sure. And the investigators were encouraged to do high-risk, high-reward science, and sometimes they sent us their reports and said, well, we tried something we thought was going to work and it didn't. And they were very good about writing the reports about it and saying, sometimes they would say, well, it didn't work and we've got 40,000 left over. We'd like to return it to you so somebody else can use it. So that's okay, and we've learned through the successes and through the failures. The mission has largely remained the same, which is to unite investigators from the level of molecules to cells to patients to populations, and leverage the fact that we have all of those represented, but the scope has been narrowed, as I described before, as part of our 2022 efforts to engage the stakeholders and see where we're headed. So the redefined scope as of last autumn is to focus on microbial contributors to human health and disease. We have 3 themes. One is pandemic threats, and many of the people in the room are working together to try to identify how we can align with the Canadian Biomanufacturing Research Fund. There are ongoing letters being written so that the next time we get hit, we are not looking around for masks and gloves, but we actually have the capacity to go ahead and try to test the new drugs, try to develop the new diagnostics, and be part of the effort to find the new vaccine. We have antimicrobial resistance. Dao Nguyen is leading the center. Dao, you're somewhere in the room. Thank you, Dao. And she's brought together 50, 60 investigators in McGill with a shared interest in AMR. Again, all the way from people looking for drugs in the lab to looking at better ways to manage antibiotic prescriptions in other countries. So a huge range, but with the focus on AMR. And then we have microbes that shape human health, and Ira King, who's the head of our microbiome, our Center for Microbiome Research. Ira, I saw you earlier. I know you're in the room. There you are. Thank you. Ira has been spearheading that. So now we have a redefined scope, although we still have that same vision that we want to be able to span and in not only different parts of the Faculty of Medicine, but we have members from the Faculty of Engineering, from Faculty of Science. We've even given grants to people in the Faculty of Law for looking at policy aspects of the research we are doing. So what's the challenge ahead? So this is what it looks like off the starboard of a sailboat heading into Positano. And when you're sailing into a town like that, you want to make sure you have a plan of how to get to the harbor. Otherwise, somebody will be screaming at you in Italian that you don't know how to park your boat. And so you have to kind of have a little bit of a plan and try to figure out, where am I going to be 5 minutes from now? Where am I going to be 15 minutes from now? And this question is, of course, where are we going to be 5 years from now? Many of you have had a job interview, and at the job interview, somebody like a dean looks at you and says, at the end of 5 years, you look back, how will you define success? What will be your measure that you did your job well? And we all go, well, good things will happen, and everybody who comes to my office will thank me. And then they go, yeah, you're unprepared. So I've been trying to think also, it's nice to say what success looks like, the first thing is you have to just say, well, what defines success, and who makes the definitions of success? And I think different people in the room have different definitions of success, and it's also dependent on different stages of your career. There are different metrics, but ultimately there should be outputs. I've been thinking about this a bit, but it's a very rough diagram, so people can help me next time I work on it. But early on in your career, when you try to get your FRQ Junior 1 or Junior 2, you sort of say, I want to get funding so I can get publications, so I can get more funding, and you feel like you have productivity. And if you keep on ramping this thing up and there's more and more dollars and there's more and more publications, you can say you're really productive. In some ways that's true, and you have an operating unit. In some ways what I'm showing, I think, is a fuel cell. And to a chemist it could be the Krebs cycle, where you have the energy kind of generating, but then you ask yourself, beyond metrics, are there outputs? Are there things that get spit out of that fuel cell that land somewhere that can make a mark? And here's a non-exhaustive list of things that I consider outputs, in part from the work I did before MI4, but hopefully the kind of work that can apply elsewhere. So I think you can have new concepts, you could have new products, you could have new policies, you could have any of them, and there's obviously other things you can say are outputs. So here's a concept at the top, written by Maz Divangahi. These are all examples from tuberculosis, because I know a little bit about TB, so I can use examples there with some confidence. Maz says, there's a bunch of people out there in the world who think you need T cells against mycobacterial antigens to make a vaccine. And he says, let's write an article to say, is that sufficient? Is that enough? Are there other ways? Should we be looking elsewhere? Galit, who you're going to hear from later, was also part of the group of people saying, well, what about other things? What about antibodies? And the idea here was that a lot of groups were saying, we can make the lymphocytes against mycobacterial antigens to a level 7, and we're now going to try to make an 8. And somebody's going to say, I'm going to make a 9, and eventually, like in Spinal Tap, somebody wants to turn it up to 11. And Maz is the kind of person who says, what if we're doing the wrong thing? And what if we can provide protection by getting rid of these lymphocytes entirely and show a completely different mechanism of action? And can that lead the field in new directions? To me, that's a new concept, and with a group of other investigators has really changed the field of TB vaccinology in a way that we cannot predict right now where it'll head. But it has certainly opened up new avenues. Here's an example of a new product. Very simple product. You know, sometimes a product is a $10 billion blockbuster, and sometimes it's something that's a non-commercial sharing entity. All around the world, there are PCR machines to detect TB, and you have to say, how do all the labs all around the world know the machine is working? So our lab developed a BCG strain which is safe, engineered a rifampin resistance into it, so that the technologists in Kujarak, the people in Vellore, India, have a control that they know the machine is working, so that they can then serve their community with that little product that's now shared around. And another new thing would be a new policy. My colleague Dick Menzies developed a 4-month protocol to treat preventively TB instead of 9 months. So there's better adherence, there's less side effects, it's become the global norm. That is the new policy. So when I think about where are we going to be in 2028, I hope for all of the things we're doing, we're going to have new concepts, new products, new policies. But what will be most exciting if we have completely new directions. Things that we can't even imagine today, where we're going in new spaces. The people in the room here are going to say, well, what about if we go there? That'll be when we can really define that we've got success. So with this, I'd like to thank you all for being here. Somewhere up here in the top left, I don't know if I can use the mouse, there's a picture of the original launch gift from 5 years ago, and many of you are in the other pictures here from many of the other events, and really appreciate you here, and looking forward, of course, to having the cake and the champagne to celebrate this event. Cheers.
Well, I'm to say some words and then do a toast so you can have your cake and drink at the same time, but I'm going to reverse that order, and with that wonderful overview, Marcel, propose a toast to the next 5 years, full of wonder, discipline, great aspiration, and the ability to take failure and understand you can learn from it, and the just drive through on the joy that comes from doing something that really is globally important. To the next 5 years of MI4. Cheers. Cheers. So you can also eat your cake now. Let me just say a couple of words very quickly. David, I want to recognize you as well. Colleagues, if I can have your attention, I know the cake and the drink are really exciting, but if I can have your attention just for a few moments. Like Marie, I refused the opportunity to get involved with MI4 And David said, just go meet with Marcel and Dawn, and then say that you don't want to get involved after that. And it worked. You've been fantastic, David, as a dean, as a vice principal for health sciences, as a colleague, just a cherished member of not just the Faculty of Medicine, but the university. We know there's a lot of future ahead of you, but I'd like to just sneak in another toast to our unbelievable David Eidelman on this special occasion. So I will just also say a few words about MI4. When I decided to switch direction in my career back in 1979 and go to the States to do a PhD in epidemiology and biostatistics, in September of '79, one of the first lectures I heard in our doctoral program was from the great Abdul Omran, infectious disease specialist from Egypt, who said, the age of infectious diseases is over. You will be epidemiologists of man-made diseases. And the whole program had been forecast, redesigned as cardiovascular, cancer, occupational health, psychiatric, on and on, all the different human-made sectors of disease and illness that you might imagine. 3 years later, as I was graduating, the first case of AIDS came on the scene. And we know what happened since then. TB's resurrected, even polio has been, new cases of polio have arisen and so forth. So to those of you who have understood through your careers how important the general fields of MI4 are, and to those of you, David, Marcel, Dawn, Marie, and all of you who've come together from the science and clinical side, and then Paul Marchand for your tremendous insight and generosity in saying you're going to start up this enterprise. We just know epidemics come and go, pandemics come and go. And with all the changes happening in the world, mobility of populations from the southern hemisphere to the northern, back again, climate change, on, you know all of this. We are facing a future where the work of MI4, you knew it when you started in your careers that led to you being able to participate in MI4, but much of the world hasn't known how important coming together around the MI4 themes is for the health of not just Quebec and Canada, but of course people everywhere in the world. And what I love, I have to say, as former principal of McGill, is that you have, understood that we have a critical mass of interdisciplinary, clinical and scientific capacity and interest and commitment here at McGill that is really comparable to none other anywhere. And I know Marcel doesn't like, words of, kind of fancy, flowery words and terms that don't have substance. But I would say with all understanding of the substance behind what you all are doing, what's being led under the umbrella of MI4, we are acknowledging the strength of McGill. We are celebrating the strengths of McGill. Paul, you and donors who you've brought along with you understand the strength and dedication and talent here at McGill. And I just have to say how proud I am. And I would say that we should celebrate our successes. We should talk about the learning from the failures, and we should not be afraid to say what we're doing is unique and will continue to be unique going forward. So I salute all of you. And so another toast, a third toast. And then I'm going to introduce our wonderful guest speaker. To all of you, to MI4, and to an aspiration and a commitment and a common engagement in the work of MI4 going forward. So with that, let me introduce Galit Alter. Some of you will know her. She's an extraordinary person. A lot of fun, a tremendously positive attitude with what she does. And I said, tell me something that isn't on your impressive bio. And she said, well, I came to McGill. All of her degrees are from McGill. I came to McGill in '95 and right into the middle of the referendum. and right around the beginning of the height of the AIDS epidemic. And she said, those two forces, both historic, have shaped me in the work that I do. And that has taken her today to be the vice president for research in immunology at Moderna, and a tremendous scientist and organizer of science, and just a wonderful person. So we're proud of you as an alumna. We're proud of you as a member of the advisory group of MI4, and so happy to have you coming and speaking today. So please join me in welcoming Galit.
Thank you all. Thanks for the introduction, and it's so wonderful to be home. It really does feel like home, seeing my PhD supervisor, Nikki, and seeing so many old friends and new friends, and just being back again to discuss science with all of you, and really to celebrate MI4, which I think I heard about as it was just being kind of grown as a little seedling, and just seeing the incredible impact that it's had, not only scientifically, but also by creating this community that is committed to ending global epidemics and infectious disease. So that's really just a wonderful moment to be here today. So what I thought I would do for my presentation, because I've got 45 minutes, and for those of you who know me, I'm gonna take you on a whirlwind tour. For those who don't know me, brace yourselves. But what we're gonna do is we're gonna have some fun, and what I'm gonna try to do is give you a couple moments of just showing you how immunology changes the way we think about vaccines. And I'm going to convince you with this talk, hopefully, by showing you how immunology teaches us that 3 respiratory viruses are controlled by the immune system in completely different ways, even though they all come through the exact same place. And I'm experiencing one of them right now, or I'm coming off of it. So I can tell you that I know which cells are involved right now. But I think that the most important thing is I'm going to hopefully convince you of why I made the move and basically crossed the street from Harvard University to Moderna, and just hopefully inspire you to think about the possibility over the next 5 years as we build a plant just 10 minutes away from here, right, that there are possible partnerships where we can accelerate medicines based on mRNA together as a community. And that's, I think, the best part of being here today and thinking 5 years to the future. So I get to do fun things now where I can do reverse disclosures, where I'm going to show you everything today, which has never been done at Moderna, but it was all done while I was at Harvard, and it was in collaboration with 3 other companies. So I'm going to do the reverse disclosure of just telling you that this was all done with 3 wonderful partners, J&J, with I think it was Sanofi and AstraZeneca, each one with a different pathogen I'm going to tell you about today. So the immunology that I've been kind of obsessed with over the last 20 years, really inspired, I think, by Nikki, who kind of told me, like, challenge the dogma, right? Like Marcel and Maz, it was like, okay, everyone's saying it's got to be this cell type, it's got to be antibodies for HIV, and Nikki said, no, it's T cells, and we're like, okay, so we went off and started doing T cells when people weren't really thinking so much about T cells. But where this story begins is really with this concept that as vaccinologists, we're kind of trained to think that the most important antibodies a vaccine induces, right, that provides protection are those that can bind and block infection through a mechanism we call neutralization. Well, that is true. Neutralizing antibodies are critically important, but the majority of antibodies that we make after infection or after vaccination have a completely different purpose. They have nothing to do with binding and blocking infection. And of the billions and gagillions of antibodies we make every single second from our humoral immune system, they're actually positioned to do something completely different. Their job is literally just to detect foreign material. And once they detect that foreign material, they serve a critically important function. They basically tell the immune system that something is wrong with this aberrant foreign thing in the system, and then tells the immune system exactly how it should eradicate anything to which that antibody is bound. So 99.999% of the antibodies in your body are doing this, and those things are critically important. We know they have an incredibly important impact in controlling infections. This is just not how we've been thinking about vaccines. So we know this is important for clearing autoimmune cells, for getting rid of tumors. We know this is important in clearing apoptotic bodies. It's also critically important in clearing infection. And so what we became obsessed with about 20 years ago was thinking about how can we build tools that allow us to interrogate all these other functions of antibodies, begin to understand how the immune system leverages those functions of antibodies, and then start making vaccines that can purposefully induce those responses in a targeted way so we can maximize protection against infection and disease. And that really was kind of the essence behind the development of a tool that we now use to essentially inform vaccine development. So the idea is to think about all the rest of the antibodies and how they contribute to clearance of the pathogen. Now, what these antibodies do is once they mark either the virus, I'm sorry, I don't have a pointer here, but if you see the virus on the top or an infected cell, they can then recruit in monocytes, macrophages, complement, neutrophils, basophils, mast cells, whatever you can imagine in a tissue, and provides them with instructions to kill. And the way that antibodies do this is very simple, right? The antibodies have the specificity on their Y-ends. They recognize the pathogen, right, because they have an infinite capability of recognizing different targets. But their back ends, their constant domain, also changes very rapidly and interacts with immune receptors called Fc receptors that are present on all immune cells in the body and even many non-immune cells. And in humans, we have lots of different kinds of these Fc receptors that are expressed in different combinations on different immune cells. So the immune system has this incredible diversity of opportunity to leverage immunity to drive different kinds of functions. And what happens is every time a B cell makes an antibody, it has to make a decision on how it wants to shape the back end of the antibody, either by protein changes on the right with subclasses or glycosylation changes, and that then leads to different functions: inflammation, anti-inflammation, clearance, killing. It can lead to all these different functions. So to begin to understand how these immune responses diversify in populations, we began to think about how we could use systems biology, machine learning, and AI to begin to tackle the problem of how is the body using antibodies to fight disease. And so we built a tool we call system serology, which essentially is an antibody omic tool. And the idea here is that we can interrogate plasma or any kind of fluid from people, and we can run it through different functional characterizations on the bottom, or we can look at different biophysical features of antibodies, asking what subclass, isotype, glycan, Fc receptor does it bind to, and how does it arm different immune cells to mediate those functions. And then we capture a lot of data, right? And that's shown in the heat map. So the way to read these heat maps, just to give you an idea, is every row is a different person. Every column is a different data point. And the only point I like to make here is that no two individuals are ever the same. Every individual in this room will have a different profile of their antibodies to flu, to RSV, to every single pathogen you've seen in your life, and that's dictated by when you were last vaccinated, when you were last infected, what you probably eat, how well you sleep, whether you have a twin or you have a brother or a sister or kids. So everyone has their own proprietary profile, but you can now use machine learning to look for profiles and patterns that are associated with good outcomes or bad outcomes, and to use that to develop mechanistic hypotheses that can then be used to develop new drugs, vaccines, or diagnostics. And we've used this over the years to tackle many different pathogen profiles, including TB, HIV, malaria, Ebola, RSV, pneumo, Shigella, and the list goes on. And so the point to all the trainees out there is that this is an incredibly flexible platform that can be used for any disease of interest, even non-infectious diseases. So what I want to do now is I'm going to just show you 3 applications, just to give you an idea of how immunology can be incredibly different across different pathogens. The immune system fights 3 different respiratory pathogens in 3 completely different ways. And by just understanding this nuance of how the immune system uses different immune cells to fight these different viruses, we can now think about how we can design vaccines that can essentially target those particular immunological mechanisms so we can essentially control and curb pathology associated with those diseases. So the first disease I'm going to talk about is RSV, and this was a study that was done by a very talented postdoc in the lab, Yannick Bartsch, and this collaboration was done with J&J at the time. So RSV is a hidden epidemic. We don't talk about it all that much. Over the last year or so, we've heard a little bit more about respiratory syncytial virus, but when I ask my parents, do they know what RSV is? They're like, is that something at the CVS that I can buy? Or at the grocery store? Is that like a new kind of like cereal? So they don't really understand what RSV is, but respiratory syncytial virus clearly causes respiratory infections. And what's fascinating about RSV, just like with COVID, is that you can be infected 1 year, and the next year, you can get reinfected again, right? Immunity lasts about 7 months in a given population, and people become newly infectable every 2 to 3 years, even if they have very high levels of neutralizing antibodies. So something else is likely to be important: control of RSV. And it's incredibly important, obviously, as a healthcare burden in the young and then the very old that are disproportionately affected by this particular pathogen. So RSV is a very interesting RNA virus, and it has 2 proteins, 3 actually on its extracellular surface. The major protein of interest is called fusion protein, and it's the protein that essentially allows the virus essentially bind and then enter into the target cell. And this fusion protein is less variable. It's an important target for vaccines. And so what J&J did a few years ago was essentially package RSV-F as a stabilized molecule in Ad26, just like it did for its COVID vaccine, and then administer it to humans and then challenge them in a controlled human challenge model, which is the perfect human experiment. And then they basically looked to see whether or not the individuals became infected and if they developed symptoms. And the question was, could we take this plasma from these individuals on the day of challenge, and could we predict who's going to be protected or not, and learn from this to understand what matters for protection against RSV. So we captured lots of data in the RSV-specific immune response. And so this is just the same kind of data that I showed you before, a heat map showing all the different flavors of antibodies that can be induced in a given individual. And just to give you, again, an idea here, every row is a different individual. Every column is a different data point. You have the profiles on the left that are before vaccination, so everyone has pre-existing immunity to RSV. And after vaccination, you can see selective induction of immunity only in the top here in people who received the vaccine. So you see that darker color coming up in those individuals. But of course, lots of things are coming up, so what matters, right? Is everything important, or are just certain features of the immune response important? And what does that tell us about how we fight RSV? So we looked at typical univariate analyses. We asked, okay, is the vaccine inducing IgG1s? That's the predominant antibody in the blood. And we see very nice induction of IgGs in the vaccinated individuals in the yellow compared to the purples who did not receive the vaccine. Not a lot of IgMs, that is a naive immune response. So not a lot of new B-cell response. This is a recall immune response. And not a lot of IgAs, right, which is a mucosal workhorse that we think is important against RSV. So this vaccine is inducing a heavily recall-dependent IgG response. We also looked at a number of functions, and we see all these beautiful functions coming up. This vaccine is inducing monocyte phagocytosis, neutrophil phagocytosis, NK cell activation, induces viral neutralization, tons of immunity coming up. But what matters against RSV? So the individuals were challenged, and we basically tracked infections in this population. And you can see here in the purple are the placebos. So you see this gradual infection of nearly every one of the vaccinees. So this was a dose of the virus that would guarantee nearly 100% protection in that population. And then you can see in the vaccinees, we got the majority of individuals were protected, but not everyone was completely protected. And what's important is knowing this is a recall response, we thought, let's take all the data and ask, are there profiles that are enriched in people who are protected that are associated with differential control of the disease? Now, what's important is that both just looking at binding antibodies as well as neutralization antibodies can predict who's going to be protected with pretty good significance, with about 80% confidence. But neutralization was not more powerful than binding. So that told us that something else that antibodies might be doing might be important. So we asked, well, what is important? So we can do machine learning. We can basically take the data and ask what really separates out the two groups, people who are protected from those who are not. And in this dot plot over here, what you can see in the scatter plot, is that individuals in the coral, in the orange, were protected. People in the blue were not protected. You can see they have completely different antibody profiles in this multivariate space. This is just using their antibody data. Now, what's different about their antibodies? Only 6 features was all you needed to separate out the two groups. And the features were really quite interesting because none of them were binding titers or neutralization. So that was not the predominant predictor of protection. But instead, what we saw was that the features that were enriched in people who were protected in the orange were monocyte phagocytosis, so monocytes that are the primary fighters in the lung, so alveolar macrophages that I know Maz loves, were probably the most important cells that basically grabbed the virus and essentially eradicated it as quickly as possible, along with binding to particular FC receptors and some IgA. Now the question is, if I take those 3 features, can I now predict who in this room would become infected or not with RSV? And at least in this cohort, 98% classification accuracy. So if you make an antibody that can hit an alveolar macrophage, a particular cell type in the lung, and drive clearance of that virus, you have a 98% probability of being protected against RSV. And so understanding this proof of principle, understanding this biology, now allows us to think about how we can make vaccines that can drive these types of antibodies at a higher level that will give you the level of protection that is required to essentially stop disease and pathology. We can use orthogonal statistical methods to prove to ourselves the rank hierarchy of which features are most important. And again, what you can see here at the very top are these monocyte and phagocytic antibodies that are critical for protection. Now, we did this experiment now in a second virus I want to just show you quickly. So I'm going to skip over this part here. Excuse me. I'm trying to get through. So we ask the question now is, what if we take a different respiratory virus? Let's take flu as an example. Do we think now for flu that the same mechanism of protection, monocytes at the barrier as soon as you come in the lung, is going to be the most important response to provide protection against infection? So my theory was that it would be monocytes again, because it's the same barrier, it's the same compartment, it's the same type of immune response. But what I'm going to hopefully show you is that Carolyn completely proved me wrong in a collaboration with Sanofi. Now, what Carolyn did, and I apologize for the formatting issue, but what Carolyn did is essentially collected samples from a trial that was done with Sanofi. And what they had done was essentially vaccinate a group of individuals over the age of 65 with their quadrivalent vaccine and follow them over the course of a season of flu and asking them if they became infected or not. And in the red are the individuals who controlled the disease, and you're going to see them in a minute. And then the blues are the individuals who unfortunately developed influenza over the course of that season. And we're going to compare profiles across controls and cases for multiple different features. I'm going to hopefully show you that the profile of responses that provide protection against flu are really quite different. So here on the left side, we looked at neutralizing antibody titers to the virus H3N2 that was infecting in that year of the follow-up study. And hopefully what you can see here, after statistical correction, there was no difference, although there was maybe a trend towards slightly higher neutralizing antibodies and controls, but the difference was not statistically significant. If you looked at just binding titers to flu, there was no difference between the two. So antibodies and neutralization, again, did not predict protection in this particular population. So the question was, can we capture data that can tell us what matters? So here what you can hopefully see is all the data that we collected, again, collecting lots of data. Every row is a different individual. Every column is a different data point that we collected for this particular H3N2 virus. And the only point I want to make here again is that there are no two individuals that are identical. Everyone has their own proprietary flu-specific immune response. And the question was, well, is there a particular pattern that's enriched in the cases on the top compared to the controls? And by eye, I could not see it. So the question is now, can we apply some type of artificial intelligence, some machine learning tools to say, can we see some differences between the two groups? So what I'm showing you over here is a scatter plot of the antibody profiles in the two groups. And I want to just make the point that while the profiles are not completely separate, the blue being the cases and the yellows being the controls, what I want to convince you of is something really important about this data. The controls in this multivariate scatter, right, where dots that are close together are more similar, dots that are farther apart are more dissimilar. You see the yellows are all clustered together in the bottom left corner of the plot, really closely packed together, whereas the blues are diverging into multivariate space in all different directions. So they're doing something different than the controls. The yellows are all sticking together, so they somehow did respond to the vaccine in some sort of more homogeneous way. And the question is what did they do that allowed them to resist flu that the blues could not do? So we can basically capture the features that are locked in this multivariate signature on the x-axis, and we can show that those profiles of antibodies are completely different between these two groups. And when we ask what is most different about these two populations, what we can see is that there's 9 features of the total 250 features that we captured that differed across the two groups. The blues are enriched in cases, and these are IgAs and IgG3s that we think would be really important against flu, but they were actually enriched in cases. But the only thing that was different about the controls was one thing, that they all made flu-specific responses that were able to interact with natural killer cells, a different immune cell within the repertoire of antiviral mechanisms. So these individuals made antibodies to the HA and to the NA. They were bound to Fc gamma R3A, an Fc receptor involved in triggering NK cells, and also drove to enhanced level of NK cell killing. And so what these guys did was they somehow made a vaccine-induced immune response that could leverage a very specific immune cell that was required for killing off this particular virus. Oh my goodness, and the editing, oh, this looks terrible. Sorry about the formatting issue here. But the way that they did this is by modifying the Fc glycosylation pattern of their antibodies, and they did this essentially in a way that gave them statistically significant different levels of glycosylated antibodies in the back end. So this really showed us mechanistically how these individuals were able to make antibodies that were able to control this virus. And most importantly, we could make antibodies with these characteristics, put them into mice, and show that antibodies that could recruit NK cells were more protected than those that did not interact with NK cells. So the last virus I want to show you, and then I'm going to try to give you kind of a feel for how this all comes together, is COVID, right? Everybody, has kind of experienced this incredible, moment in vaccine development history, where we've seen a number of vaccines being developed for COVID. And so for one of these vaccines, Paulina Kaplanek, a very talented postdoc in the lab, decided to try to ask the question, is there a particular flavor of antibodies that might be important in the control of SARS-CoV-2? And this was done in collaboration with AstraZeneca. Now, what I think is really important that we saw over the pandemic is we saw that as new variants of concern began to emerge, they slipped through the neutralizing antibodies induced by the vaccines, and we saw increased levels of transmissions globally. But what we did not see was a concomitant rise of severity of disease, of cases in the populations that were vaccinated. And so we saw that the vaccines continued to provide protection against severe disease, while they did not provide protection against transmission, telling us that there was something else these vaccines were inducing beyond neutralization that might be critically important for protection against this particular virus. So the question is, well, what is it? So in this study, Paulina partnered with our good friend Shabir Mahdi in South Africa, and we received samples from individuals in the AstraZeneca trial that either became infected over the study period or remained uninfected for the entire 2-year follow-up. And we asked the question, was there differences between these two antibody profiles? Now, what's important is that the antibody binding titers did not differ between individuals who developed COVID in the pink versus those who did not develop COVID in the yellow, either to the wild type strain or to the beta strain that was circulating at the time, nor were there differences in neutralizing antibody responses, so very important. But when we went and looked at lots of antibody data in this population, again capturing every individual here, every row is a different individual, every column is a different data point, we began to see by eye with some differences in the data. So we started noticing, and you can't see this on the screen, I'm sorry, it's very difficult to see, but I'll show you in the univariate plot. There were 2 differences in the profiles, and it's shown here in the univariate plot. For the individuals that essentially became infected, they ended up having higher levels of antibodies that bound to Fc gamma R2b, an inhibitory Fc receptor, whereas they had equivalent levels of IgG1s, 3s, Ms. They also had higher levels of IgAs, again, which you think would be protective, but was not protective in this vaccine. But the one thing that was critically important is that individuals who became infected, who developed COVID-19, did not make any antibodies that were able to bind to a particular Fc receptor called Fc gamma R3b. So they basically lacked this one fundamental function of antibodies that was not present in the individuals who developed COVID-19. To prove to ourselves that this was really statistically significant at a multivariate level, we took all the data, we put it again into one of these models, and we asked, can you separate out the profiles in people who are infected, who develop COVID-19 versus those who do not? And again, hopefully you can see here, there's almost completely separate antibody profiles in individuals who ultimately developed COVID-19 versus those who did not. And it was interesting when we looked at what was predictive of these differences. There were lots of features in the orange that were enriched in the cases. They had lots of antibody responses. It wasn't that they weren't making antibodies, but they lacked one single type of antibody. They lacked antibodies that were able to bind to this Fc receptor called Fc gamma R3b, shown on the very top. So what is this receptor and why is it so important? So I told you about Fc gamma R3a for NK cells. It's important for arming the NK cells to kill flu. It has a twin sister called CD16b or Fc gamma R3b, and it's only present on neutrophils. So this is a different cell type now. It's not monocytes, it's not NK cells, it's only neutrophils. This Fc receptor has an extra little dot there, which is a glycan, which is a giant sugar, like a Christmas tree that sits on top of this receptor. So even if it's identical in its protein sequence, it has this massive sugar that makes it change the kinds of antibodies it interacts with. And so that tells you that it interacts with different antibodies, and it does not have the same signaling domain. So the question is, why neutrophils? What are these neutrophils doing? And why are these people who are resisting COVID-19, why are they relying on neutrophils? So it turns out that individuals who can bind to 3b do opsonophagocytose through neutrophils more effectively. So the individuals who were controlling, did not develop COVID-19, drove much faster clearance of virus through neutrophils. But even more interesting, if you look at the consequences of uptake, when the antibodies go into neutrophils, what was wild was that while both people who developed COVID or not had antibodies that could arm type 1 interferon type cytokines, what they did not do is the controls in the orange here never drove pro-inflammatory cytokine cascades. So they drove opsonophagocytic clearance of the virus through neutrophils that are first on board, but they did it in a stealth mode with zero inflammation. So they don't recruit in NK cells, they don't recruit T cells, they don't recruit macrophages. They basically clear the virus completely quietly and now allows the immune system to essentially get rid of the infection, but not develop any symptomatology. And we learned that exactly which glycans, which sugar patterns were present specifically on these people that could drive these stealth neutrophil functions. And more interestingly, we could show that antibodies with these very interesting sugar modifications could actually drive completely different transcriptional cascades in neutrophils in the cases compared to the control. So they're arming neutrophils with completely different transcriptional signatures. So what I want to leave you with is this idea, right? These new immunological tools, these omic-like technologies that allow us to look at the immune system at unprecedented depths, now shows us that there's much more to how antibodies are fighting viral infections. And just as 3 examples of 3 respiratory viruses that all come through the same passage, hit the same cell types, hit the same mucosa, 3 fundamentally different immunological mechanisms are at play in controlling and killing those targets. We see that neutralization with monocytes is probably important for RSV. We think that influenza depends on neutralization and NK cells. And SARS-CoV-2 requires neutrophils with neutralization. And so understanding this combinatorial dynamics, understanding this biology, now tells us how we can begin to design vaccines now with purpose, with rational immunological information in mind. So with that, hopefully you can understand why it was that I became completely excited when I thought about how amazing would it be if you can take this information and literally within months be able to develop vaccines, that could tackle or drive and train the immune system in just the way that you think is critical for driving protective immunity. And this is really what brought me to Moderna. So I'm going to give you just a few slides on really what it is about the Moderna technology that is really so exciting, and to hopefully open up discussions about potential collaborations in the future. So I think the COVID epidemic really showed the power of the mRNA technology, but it's not a new technology. It's been around for at least 10 years, if not far before that, where we were working on developing ways of essentially delivering software, RNA, to cells to program them to fight tumors and cancers and other types of diseases. But it was in the context of COVID-19 that I think we saw the power of this technology, which is incredibly quick, it's incredibly flexible, and relatively easy to essentially disperse to the entire population. And the idea here is just with the COVID-19 vaccine, and this was really a beta 1, a test, I would say like a 1.0 effort. It went from sequence to actual product in about 11 months. And now those timelines have been contracted significantly, where from the time you have a sequence, the protein is designed, to the point where you can synthesize the RNA, vial it, and deploy it, really we're somewhere living in probably the 6-month time frame. And so this is really what we're talking about, probably even condensing that further. And what makes the RNA technology, at least with Moderna, so special is that it's really a very simple 5-component system. There really are only 5 ingredients to this particular vaccine that makes it really as effective as it is to date. What we say is that mRNA is a drug product, right? That is the sequence, the software we're delivering to cells. And that software has been designed over the last 10, even before 15 years, to essentially develop the right structure of the RNA that gives us superior production and translation within the cell. And that's really the code that we're trying to deliver with the actual vaccine. And in addition to the mRNA, which we call the drug product, there are 4 additional components. There's an amino lipid that's ionizable. So this lipid is critically important for allowing the mRNA to getting the lipid nanoparticle into cells, but more importantly, allowing it to escape the endosome. And that's really the power of this ionizable lipid. We have sterols that stabilize the outside of the actual target, including the phospholipids. And then we have a PEG lipid that sits on the outside that many of you probably heard about, which essentially really provides stability once that actual lipid nanoparticle gets into the system, really protecting it from other cells or other types of components within the serum that can attack the lipid nanoparticle. So it's a 5-component system that essentially allows you to get in, release, and then translate. And the idea is really quite simple. We go into the muscle because it's a contained target site where you can deliver a known volume. The delivery of the vaccine to the muscle allows disruption of the muscle, recruitment of immune cells that come in and essentially capture most of the drug product. So if you look at where the drug product is, it's almost exclusively found in the recruited macrophages that are coming in because you've disrupted that muscle fiber environment. Those cells then run, obviously, to the lymph nodes where they trigger adaptive immune responses and essentially lead to translation of the protein within the macrophages that then can drive T and B cell responses. And I think what makes this mRNA technology so powerful is really because it essentially uses the exact same pathways that are typically used by viruses to essentially infect translate, and induce immunity. So if you think about a viral life cycle, the virus comes in, the virus triggers a number of different kinds of innate immune responses, then leads to inflammation that tells the immune system that something's going on. You've got to drive an immune response. And then the virus then produces its proteins and then essentially is able to then make new progeny of itself. Now with the mRNA, we have no progeny, but we're triggering similar pathways, although not all the same pathways. We're essentially using the same endocytic pathway to come in to trigger at least enough inflammation so you can trigger an adaptive immune response, but then the translation occurs and the mRNA is degraded and gone. So there's no progeny after the fact. Now the key learning from making this mRNA vaccine most effective was learning how to temper the innate immune system, the innate immune response. If you're like a virus and you drive too much inflammation, you kill and restrict translation. So you have to figure out what parts of that innate cascade are important. Which ones do you want to ramp up? Which ones do you want to diminish so you get enough translation so you can drive adaptive immune responses? And so in that process, over 10 years, there have been multiple key learnings. One of them, I think, has been publicized very widely about the importance of changing nucleotides. They don't trigger particular innate immune responses. So everyone's heard about the change in the pseudouridine, which allows you to evade TLR7 recognition, or which is a key modification that Catlin and Drew developed many years ago that led to the mRNA design of many of the products that are out there right now. But there's additional changes that we made to the RNA by eliminating these short double-stranded RNAs that can trigger innate immune receptors, as well as recondition and model and shape and build our RNAs so they don't fold back on themselves. Because if they fold back on themselves, they form double-stranded RNA, which our body does not like to see. And so by essentially diminishing and engineering our RNAs to eliminate double-stranded RNAs and to eliminate refolding back on itself, in addition to addition of the pseudouridine, we can now control the level of inflammation and we can improve translation, which might be why the Moderna vaccine induced such whopping immune responses at a global level. And just to leave you with one last thought, as you think about the future, at least within Moderna, there are at least 36 programs that are moving forward to clinical development. And this is just here kind of a smattering of some of these programs. In the blue are all the prophylactic vaccines. We have a number of cancer vaccines that you may have heard about, melanoma moving to phase 3, but a number of additional checkpoint vaccines and others coming forward, as well as cytokines that are being used for immunomodulatory activities, as well as a number of additional targets that we're going after to essentially see if we can try to drive therapeutic activity in rare disease. So it just gives you an idea of some of the programs that are moving forward, and just to give you, again, this opportunity to imagine what is possible, right? That this is really an idea where Moderna is trying to partner with the academic community to think about ways that we can begin to move more quickly to drive protection against different diseases. And we also have a number of enzyme replacement therapies that are moving forward for rare disease. So what I want to leave you with is this idea that the COVID vaccine, we've seen this, incredible speed, 11 months to a product to provide protection against a disease. But now we're moving into a new phase of an endemic life where we're living with this pathogen. And so really what we're living with right now is this idea that we can essentially move with the pathogen even more quickly. And so there are multiple different programs that are moving forward right now. On the top, which is basically, you've already seen the variant vaccines that are moving forward, where we believe that in about 4 months, 5 months, we can essentially bridge the gap and to get those variants into people's arms as quickly as possible, so we can make and mount the most effective immune responses, hopefully in record time, so we can get people the right type of immunity to provide them with protection. But the option number 2, which is also coming, is ability to start to combine vaccines, where eventually we're going to be able to do combinatorial vaccination across multiple respiratory vaccines, or enteric, or bloodborne. The idea would be is to try to think about ways that we can condition the immune system and give people one less needle stick in their arm, because who really likes a needle in their arm? Okay, so with that, I'm just going to thank the people who did the work, and I thank you for your attention. It's always the champions, the people who actually do the work, that always deserve the most recognition. I just get to be like their cheerleader. But Yannick did all this work on RSV. It was really phenomenal with our colleague Rosemary Bastian at Janssen. Carolyn Bruder did all the work with phenomenal colleagues, especially Janet McKelney from Canada here. She was, I believe, in Ottawa. She was wonderful and unfortunately passed away last year, with Harry Cleantis at Sanofi, and Paulina Kaplanik with our friend Shabir Mahdi and his group in South Africa at the WITS. And I thank you for your attention, and I'm open to any questions.
Thank you, Galit. That was wonderful. I'm just looking for volunteers with their hands up. Ideally, students go first. And I know there's some students from my lab, so I don't want to put them on the spot. But any trainees, fellows like Lauren Schweitzer, not to name Lauren, have the first dibs. There you go, Lauren.
Can you hear me? Yeah. Thank you so much. That was an excellent talk. So you mentioned when talking about mRNA vaccine and designer vaccines that everybody responds differently to a given antigen. So how do you develop a vaccine that's going to be distributed globally, like the SARS-CoV vaccine, that's going to develop exactly the antibodies you want it to develop when everybody has their own proprietary, I suppose we could say, antibiome?
That's a really good question. So I think what was important, at least from the flu story, I don't know if you noticed this, Lauren, but what was important from that study was that everyone had their own overall antibodyome, but there was still a flavor that made the controls different, right? They did something that was always the same that made them somehow special. And that was the ability of those individuals to make those NK cell triggering antibodies. So the idea is I don't think vaccines have to homogenize. Right? I don't think that that's ever going to happen. We have to accept the fact that we are all unique and we're all snowflakes and we're all going to have the same type of unique immune responses to any target or antigen we come in contact with. But what we want to be able to do is to try to promote higher levels of those specific flavors of immunity that we think are going to be the most impactful and the most important. And so using these types of mRNA vaccine design approaches that allow you to essentially deliver the antigen with the right signals that drive that flavor of immunity is really what's important, right? And we've always kind of held this dogma in our minds that what is important is those neutralizing antibodies. And there's no question that neutralizing antibodies are important, but maintaining high enough neutralizing antibodies in the upper respiratory tract for a long period of time is such an incredibly difficult, immunological challenge. But trying to think about how you can mount high enough responses of these functional antibodies that can then temper the activity of the virus or the pathogen in the lower respiratory tract, that is something that we can tackle as an immunological community using these mRNA vaccines. I think that that's really the mission, is understanding what is that flavor and inducing that flavor and not trying to homogenize the entire immune system, because that's not going to happen.
And so on that point, what about mucosal delivery versus intramuscular delivery?
Yeah, so this is a really interesting debate, and we hear these discussions all the time. There's no question that mucosal delivery will drive different immunological functions because you have different types of flavors of antigen-presenting cells at those particular sites. The problem is that mucosal delivery is very difficult from the perspective that, if you put in 1 ml or if you nebulize a certain volume of material into one person's lung, our mucus is completely different. Our ability to penetrate down to the epithelial barrier to drive those types of immune responses is completely divergent. So you never have complete control over the volume of the material that you're delivering. Now, what is possible, though, is the possibility of delivering antigens to the muscle or other places that can now recapitulate those types of flavors, potentially, of signals that are associated with the nose. And so there are ways that we can think about engineering the system, right? We don't have to think in a linear way anymore. We used to think, I think, in a dogmatic way that if I want mucosal immunity, I have to go to a mucosal site. Well, what I hope that I can convince you of today is maybe I don't have to do that. Maybe what I can do now is I can go to a different, easier to access site, but I can trigger some signal that kind of tells the immune system that this pathogen is actually happening at a mucosal barrier, and it's now going to program a response that would be similar to that you would have induced there. So I think these are two different dimensions of ways that we're thinking about the problem, but there's no question that there is a big momentum to think about how we can break the problems of the access in the upper respiratory tract.
Thank you.
I think I see Ira's hand going up.
Hi,
Galit. I just noticed that you see transcriptional changes in the neutrophils that are expressing the Fc gamma receptor 3b.
Yep.
But I noticed that it's a GPI anchored receptor.
Yes.
So I can understand why that would not lead to a hyperactivated response or a silent uptake. So how does it work?
Okay, this is deep immunology. So imagine, right? Imagine this world where in an immune response, you don't have a single antibody touching your virus, but you have an entire complex, right? Your virus is now coded in hundreds of antibodies, right? And even more magical, you imagine, it's almost like a lattice of antibodies tethered to different viruses. Now you have all these Fc domains that are now triggering and binding to different Fc receptors on the neutrophils. And so the difference between the 3B binders and the non-3B binders is the types of Fc receptors that are clustering within that synapse. So what happens, right, when you don't bind 3B, then all that goes in the synapse are Fc gamma R2A, 2C, and alpha receptor. When you go on the 3B, though, what you're getting is a different combinatorial dynamic of Fc receptors. So you're still getting 2A and alpha and 2C in there, but you're getting less of them triggering those neutrophils. So I think that what's happening, right, is that you have this differential quality of signaling that's happening. It's not that there's no signaling on the 3Bs, because they're signaling enough to get opsonophagocytic uptake and destruction and clearance of the pathogen, but they're not driving this crazy pro-inflammatory response. So having that GPI kind of anchor receptor in there, I think what is happening is just tempering the level of activation of that immune complex. And by tempering those signals, you're basically just driving a different level of transcriptional activation that might be essential for controlling the bug.
Bastien?
Conveniently, very inspiring. So can you give us a sense of what are the factors that you can use in a vaccine to influence the type of response? So is it the nanoparticle composition? Is it encoded in the mRNA? Could you do you add adjuvants? What are the factors that can you encode other extra protein that would be messages? So how do you do this?
Sky is the limit. That's the answer. I mean, this is really like, when I asked myself that question is when I signed. But it really, I think, it's what's important, right, is just to think about the possibility that there are things that are going to be, more clinically developable versus not developable. And there are many tricks that we have within the immune response to condition and trigger immunity, right? That's the beauty of it. And so even just thinking about, the delivery route, right? Thinking about how can I basically modulate the immune system just by moving where I'm immunizing, intramuscular, sub-Q, intradermal, right, different parts of the body. That already gives you access to different APCs. But of course, you can then co-deliver, you can modify the RNA structure, you can modify the LNP, composition. There's so many different ways that you can think about using this, essentially try to mimic or drive the response towards different directions. Right now, I think that what we have is a product that is, optimized for inducing very high levels of immunogenicity, but perhaps it's not yet at the point where we're saying we want to drive this response or another. But the cancer vaccine, on the other hand, goes in a completely different direction, right? Because now what we're doing in the personal cancer vaccine setting is we're sequencing a person's tumor, and then within 8 days, we're essentially putting that into a dirty lipid nanoparticle that triggers immunity, and then we put that into the system, and we're asking the T cells to now make responses directly to that own person's proprietary peptide, tumor peptides, and to really respond to this very robust immunogenicity that's driven by the LNP. So just to think about, right, that the possibilities right now is, you think about it as this is kind of 1.0. And the question is, where can you go next? But the possibilities, I think, as you see like hundreds of companies popping up every day with RNA technologies, are really exploring this incredibly vast opportunity of how we can basically hijack and use this to basically mimic different types of immunological responses. Okay, I see he's checking his time.
No, we're okay. One more question there? Behind Matt's first. Sorry, I don't know your name. Yeah, we'll get you the microphone. And then Mads gets the last question.
Thank you.
So
Thank
be
you
careful.
for the talk. My name is Roseanne Tom, class of '87. So my last class was about 36 years ago, and I learned that RNA was always fussy, degradable, oxidizable. You couldn't put it on the counter and it would just disappear, and everything had to be ultra clean. And good luck trying to extract it and manipulate it. So now, obviously it's been like now the last 15, 20 years that we're able to manipulate mRNA, RNA, and transfect it, use it as transfectable material. What is the secret soup that makes the RNA now so tough that it's manipulable, and now we're making vaccines as opposed to DNA?
So I don't think there's a secret sauce, honestly. I think that everything you said is absolutely true. RNA is an incredibly unstable molecule, right? That dogma has not changed. That biology has not changed. But the chemistry associated with understanding how RNA can be stabilized within the body, I think, is what taught us about how we can now make lipid nanoparticles that can mimic that biology. So just think about this right now. We constantly have cells communicating with each other every single second of every minute. You've got bacteria communicating with your brain cells as you speak. And the way that they do that is by releasing exosomes, right? Every single cell in your body is like releasing little bits of information, and it's basically creating this massive network of information that's communicating across the entire system, right? You have this incredible highway. And those lipid nanoparticles, these little exosomes, are pieces of your lipid barriers that essentially carry in them nucleic acids or other types of signaling molecules. So your body naturally is constantly stabilizing nuclear material to send off and to shoot information to another cell type, so that you can empower yourself to fight any disease you come in contact with. The difference now is that what we realize is that there are particular lipids, there are particular chemistries that work better with RNA. And by learning from the natural immune response and understanding how we naturally are able to create this information highway and create these lipid nanoparticles normally in our body that go from our toe to our head, right, and can live that stability, we can now mimic that and we can optimize it further by basically doing combinatorial chemistries. So that's really all it is, right? It's that kind of realization that, yeah, RNA on the table is useless, but our body is using RNA every single second of every single day to create information highways. And so it's just leveraging that biology now in a vial that's allowing us to make these new lipid nanoparticles that are essentially just mimicking what we normally do every single day.
Thank you.
Maz,
All right,
question.
Maz. Yeah, exactly.
Make it a hard one.
No, it was a beautiful talk, Galit, as usual. Really, really enjoy very much. I just have a quick question. So all these cells that come in, like these reprogrammed neutrophils or NK cells, eventually they have to come from the bone marrow. So is the idea is that some of these antibodies reprogram hematopoietic stem cells to generate that, or this is happening at the local level?
It's so selfish. No, but this is a good question. I think, but it's very selfish, right? Now, so to answer your question, Maz, the first answer to your question is, I think your question is irrelevant for this particular question. And I'll tell you why, but I'll tell you why I do think that what you're getting at is really important. So the reason it's irrelevant is that the responses that are required to drive this type of immunity is happening within hours, if not days. It's too fast for the antibodies, I think, to be driving some sort of like bone marrow change in order to get those cells on board in time to clear the pathogen as fast as it's needed to ablate and control symptomatology. That being said, okay, so for this particular instance, I don't think that the bone marrow is involved here. That being said, I think what we have seen in other contexts, right, where there are infections, where there are people who naturally control or naturally clear infections, especially with tuberculosis as an example, there we do see that antibodies are doing totally non-canonical, they have non-canonical functions that have nothing to do with just purely stimulating or killing off immune cells and clearing the pathogen. But what they are doing is they're reprogramming some sort of myeloid precursor through some sort of education process that we've discussed, right? They're triggering these FC receptors and triggering these signals in these myeloid precursors that's allowing them to be more armed and dangerous when they come in contact with the bacteria. So, we think about FC receptors in, again, a very conventional way, right, here, where it's just like first responders kill, right? And you're killing through different types of soldiers or different kinds of, arms. What we're talking about with, these pre-programmed responses that give you this ability to overcome disease if you come in contact with it later, like if you've been pre-exposed to TB and then come in contact with it again, right? In that context, what it looks like is that those antibodies are now training your macrophages so that the next time they see that bug, they are much more aggressive in their ability to handle that bacteria. And so that's where I do think that there is a bone marrow involvement. I just think in the context of these responses, I think it's very much a barrier response kill. That doesn't mean that in the future we can't make vaccines that could train so you're prepared through different multiple mechanisms, not just through this one.
Yeah, but I'm wondering where are these neutrophils are coming from?
Oh,
because, they are I mean,
Yeah.
they're coming from the bone marrow. And
Yeah. I mean, at least from the mouse studies that we've done, where we've done some intravital, it's coming right from the blood, and it's not many that you need. You need very few numbers that come in directly from the vasculature that basically control and clear the virus. We haven't done a dose escalation, so I don't know if you had higher doses, if you then would need to have an efflux from the bone marrow. But for these, because the MOI is relatively low, it just looks like it's just coming from the blood. But it's a good point.
All right. Well, that was fantastic. There was so much information that I'd like to do a quick mashup to make sure I understood what happened. We're all unique snowflakes. Stealth neutrophils are good, and Maz's question is completely irrelevant. Is that a good summation? Thank you very much, Galit. I'll call upon Marie for some closing remarks.
So indeed, thank you very much. I don't know if you're going to get invited again, but it was really a fantastic talk. So I'm not going to keep you long. I just have a few small things that I'd like to point out. So first of all, events like this and the day-to-day at MI4 doesn't happen just between Marcel and I. We have a whole team of people who make us look good and keep us on our toes and keep us honest. And I would really like to call them out by name. So Kendra Tonkin was here earlier. She's our program manager. She keeps the boat afloat. Tasha Allende. Tasha is our senior advisor. She's at the back. Don't be shy. Lynn Derry sort of stepped up during COVID. Lynn is also she's in the back. So she was our special advisor. And finally now we have Philippe Simoas, who's our new office administrator. So hopefully you should get to know Philippe. Hopefully he'll be around for quite some time. I'd like to say that we have many, many more exciting things on the horizon for MI4 in the next year. That you should, look at your newsletters that we send out. First of all, I'm going to mention that we have the MI4 is giving out 2 prizes at the Faculty of Medicine Clinical Innovation Competition on May 11th. And in fact, Sarah Mashid, who was here earlier, has been a recipient of that award in the past. And Sarah is a bioengineer, and she really represents what we're trying to do at MI4. She's a bioengineer. She works with virologists, and she tries to really move this agenda forward. So we'll be announcing a couple of extra prizes there. The other exciting thing this year, for the 3rd time, we'll be having the Pfizer Early Career Investigator Award. Those will be launched sometime in May, and we have the pleasure also of having Vratislav Hadrava, who's the Vice President and Medical Director at Pfizer, who's been a very strong supporter of MI4 through these awards, and we'd like to thank you for that. And then the latest event that will happen this year will be our fall symposium, which will be a very heavy-duty scientific symposium. And we're happy to announce that our keynote speaker will be Akiko Iwasaki from Yale. So we really hope to have some exciting science to present to you at that time. And then the very last thing that I'm going to say, the good news is I was texted during the presentation that there's apparently a lot of bubbles left over. So please help yourself to another glass of bubble on the way out. So thank you very much for coming, and we look forward to seeing you again.